In this video, we'll be talking about the genomic analysis of COVID-19 for May 15, 2020. For the full situation report and more regionally focused analyses, please visit our website at nextstrain.org. For this report, we analyzed 5,193 publicly shared COVID-19 genomes. By comparing these viral genomes to each other, we can characterize how SARS-CoV-2 is evolving and moving around the world. This week, we give an overview of viral mutations and what they mean and don't mean for the COVID-19 pandemic. You may have wondered about viral mutations. While changes in viral genomes over time are normal. SARS-CoV-2, the virus that causes the disease COVID-19, is a coronavirus. Like other coronaviruses, SARS-CoV-2 has a genome encoded in RNA rather than DNA. Viruses infect cells because they need to borrow cellular machinery to replicate. For RNA viruses, this process is quite error-prone, as most RNA polymerases are not able to proofread and correct their work when making copies. This leads to frequent mutations in the viral genome. These are normal and expected. Importantly, the vast majority of these mutations either break the virus such that it can't transmit or replicate anymore, or don't change the virus at all because of the redundant encoding. This means that the mutations don't change the proteins and therefore function of the virus in any way. Other changes may modify a protein of the virus very slightly, but not impact the function at all. Rarely does a change in the genetic code of a virus can help it to replicate or transmit better, but nearly all of these changes still have only a tiny effect. These differences in the genomes between viruses can be used as breadcrumbs to trace the history of a given sample. Just as the people in your family share the unique combinations of genetic characteristics, closely related viral samples share unique combinations of genetic mutations. Here we show the viral family tree, with each sample colored by which amino acid is present at a specific location in the viral genome. Our example uses the gene ORF3A at site 57. Highlighting samples from New York, we see that most of the cases from New York and Europe have the same amino acid, histidine, rather than glutamine at this position. This, in combination with all the other sites in the genome, tells us that these cases are all closely related to each other. Because viral genomes inevitably change, these differences tend to accumulate at a steady rate over time. Here, the x-axis shows the date that each sample was collected. The y-axis shows the total number of mutations each strain is away from the root of the tree. While there are certainly some outliers, on average we see changes accumulate at a rate of about 24 substitutions per year. This means that if a single viral lineage were transmitting from one person to the next for a full year, we'd expect its entire genome to accumulate roughly 24 substitutions by the end of the year. As the whole SARS-CoV-2 genome is around 30,000 bases, this corresponds to roughly about 1 mutation per 1,000 bases in a year. For context, influenza would average about 2 mutations per 1,000 bases per year, and HIV would average about 4 mutations per 1,000 bases per year. There have been many reports about multiple strains of SARS-CoV-2. First, a clarification. When virologists use the word strain, it's often just as a useful label so we can all refer to the same group of samples. This does not imply any known functional difference. Separately, strain can be used to refer to viral genotypes that are functionally distinct. Importantly though, determining whether two genotypes actually are functionally distinct requires much more experimental, clinical, and epidemiological data than we currently have. A recent preprint suggests that a single mutation from amino acid D to G at site 614 in the S-spike protein may be responsible for increasing the transmissibility of SARS-CoV-2. In many geographic areas, the relative frequency of 614D compared to 614G increased over time. If a certain genotype leads to better transmissibility, we would expect its frequency to increase over time. There are, however, other explanations to why the relative frequency of some genotypes would increase. For this site, we see a number of geographic locations where initially, most viral lineages in circulation had the D allele. Later, this balance flips such that most lineages have the G allele. This could be a hallmark of G being slightly fitter than D. However, this could also just be a side effect of the natural history of the pandemic. Early on, most strains exported from China had a D allele. Later, most strains from Italy had a G allele. It could just be that this particular genotype got lucky and spread so rapidly because it got there first. There is a fair amount of debate regarding the relative merits of these two hypotheses. It will be important to carefully tease these apart, but we don't have scientific consensus on this issue yet. In conclusion, here are some takeaways from our report this week. Viruses naturally acquire mutations as they replicate. The SARS-CoV-2 mutation rate is very typical for coronaviruses. And as far as we know, there is only one functional strain of SARS-CoV-2. Lastly, teasing apart the biological impact of specific mutations is tricky. Many perceived differences can be attributed to chance and epidemiological factors. Here are some steps you can take as an individual. Practice strict social distancing, especially if you're in a vulnerable group. Remember that even if you're not super vulnerable, many people around you are. Follow these practices to protect others. Wash your hands like you just chopped a jalapeno and have to change a contact lens. Stay home as much as possible, especially if you're sick. And lastly, if you're an employer, encourage your employees to stay home whenever possible. Here are some other steps your officials can take. Some examples are to make testing free and broadly available, put strong social distancing measures in place, fund and implement extensive contact tracing efforts, and lastly, financially support those impacted by social distancing measures. We would like to acknowledge the amazing and timely work done by all scientists involved in this outbreak. Only through the rapid sharing of genomic data and metadata are analyses like these possible. We would like to note this will be our last weekly situation report for a while, though we aim to support similar reports from local public health officials. We will, of course, continue daily updates to the global and regional builds as new sequences are made available. Thanks for watching.